Normal is 60%. What is the chance that this individual would be a girl? 30%. Yeah? So you calculate the probability of this child being a normal girl in the very end. So basically, I don't know why I don't have this. So basically, these are the possibilities for this couple based on the penetrance. Instead of the chance of having an abnormal girl, 25%. Normal girl, 25%. Abnormal boy, 25%. And normal boy, 25%. This is distorted. Because of the penetrance, the chance of having an abnormal girl, 20%. Abnormal boy, 20%. Normal girl, 30%. Normal boy, 30%. Make sense? So it is not equally distributed between these because of the penetrance. Number 3. Now you see. In the following pedigree, Marfan syndrome with the penetrance of 80% is transmitted. Suppose you don't know the pattern of inheritance. I'm sure you know Marfan is autosomal dominant, but sometimes a pedigree is given to you and you don't know the pattern of inheritance. So first, you have to figure that out. So, whenever you have too many individuals affected in a pedigree, and especially when the individuals who are affected are present in all of the generations, think about a dominant one. And it is easier to exclude a pattern of inheritance than prove it. So the criteria actually, or let's say the element that excludes X-linked dominant is what? Male-to-male transmission. So whenever you saw male-to-male transmission, X-linked dominant is excluded. So you have to think about autosomal dominant, especially because almost the same number of males and females, not the same, but it is almost the same. You would see that are having this allele. So basically, this is autosomal dominant, just in case you didn't know what Marfan is. Then, in here, we have some characteristics for autosomal dominant. If the penetrance of a disease is 100%, this means whoever has the bad allele shows it. Whenever the penetrance is 80%, this means only 80% of the people who are expected to having the symptoms actually show it. 20% do have the bad allele or the abnormal allele, but then they show it. They don't show it. So, another criteria is that whenever penetrance is full, whoever is affected with an autosomal dominant trait and is not the result of a de novo mutation, the first case, so we have other individuals who are also affected in that family. So we have to find at least one affected parent for every affected individual. And I think this is what I recommend my students. Always when you know this is autosomal dominant or any dominant and penetrance is full, look for at least one affected parent for every affected child. So here we have this father affected. And these two don't have it. You see, these two are affected with the same type of disease, but they don't have an affected parent. And always remember, whenever one of the parents is not shown in a pedigree, wife or husband of someone, this means that this individual is genotypically and phenotypically for that disease normal. So his genotype or her genotype does not affect the transmission of the trait. So you have to assume the wife of this man normal. So this is autosomal dominant and this man doesn't show it. Why? He has two affected daughters, especially because he has an affected mother as well. So basically the path of transmission was from this mother to this son, So what is the genotype of this son? For sure, big M, little m. And this one, these two, both big M, little m. So the genotype of this man actually determines the possibility of transmission of the allele, not being normal or abnormal, the genotype. So when these two are affected, this individual certainly genotypically is big M, little m. So when we have a person who is big M, little m, and the penetrance of the disease is 80%, the question is asking what is the chance that this guy, 3-3, is going to be normal? Again, we would say the chance that this big M, not the little m, is passed to the offspring from this man is 1/2. The chance that this individual shows it is 80%, so it would be 40% affected. So the chance to be normal is 60%. Got it? Make sense? Very good. Okay. Albinism displays genetic heterogeneity. Some of the traits, like normal vision, normal hearing, and as well as normal pigmentation, displays genetic heterogeneity. Maybe this is a repeat for you. This means that various types of genes are involved. Either of these genes is mutant with an abnormal product, depending on the pattern of inheritance, the same symptom would be seen in the offspring. So, albinism is one of those types which displays genetic heterogeneity, which means there are different genes involved in normal pigmentation. One of them is the T gene, suppose, and T gene encodes production of the enzyme tyrosinase for deposition of the pigment in the skin cells and iris and other cells. This is gene T. And it undergoes recessive mutation. Therefore, whoever is little t is affected with albinism for gene T. Another gene is gene D, which this one produces the enzyme or produces the pigment. If I said deposition, it was wrong. So T is tyrosinase gene, which means catalyzes the product of this gene, tyrosinase, catalyzes generation, synthesis of the pigment. Gene D regulates or controls deposition of the pigment and also undergoes a recessive mutation. So whoever is affected is little d. So have this in mind. So when a person is albino, could be albino because of being affected with this type of albinism. No pigment is ever produced, so this person is albino. Or this person may be able to produce the pigment but cannot deposit this pigment in the skin cells. So it would be again albino for gene D. And very unlikely it is possible that someone is albino for both, which is very unlikely, but we have to take it into account. So we have to consider all of the possibilities. In this scenario, it says that we have a man who is albino for gene T. So what is the name of this man? An albino man who is lacking the tyrosinase enzyme. So he is little t. And married a normal woman who is heterozygous for both genes. So this is the woman. Since these are rare conditions, due to the rarity of these diseases, whoever is not having an autosomal recessive trait doesn't show it, and consanguinity is not involved, we have to assume him or her homozygous normal for the autosomal recessive allele. So now you have the genotypes of these man and woman, and yes?
Can you say why it was homozygous dominant?
Yes, because, incidence or prevalence of diseases are very important. Unless the incidence, in terms of the population genetics, the prevalence of or the frequency of the abnormal allele is given to you, which probably you would learn this if already have not. Or if we have, for example, some individuals within a family who have married together, so it is likely that someone who has married his or her cousin, both of them together have gotten a recessive autosomal allele from a common ancestor in the past. So not in this situation, which is an exception. Otherwise, two unrelated people are married. One of them is, for example, we know heterozygous for gene D and T. Suppose we know it. But this man who is albino for gene T and who seems to be unrelated to this woman and is normal, we have to assume all of the individuals who are normal for a trait which is rare, homozygous normal. So, for example, there is no reason for me to think I am a heterozygous or a carrier for hemophilia or I'm a carrier for albinism. Unless there is a history of this disease in my ancestors or this disease runs in a family, so, okay? So, basically these are rare diseases, so that is why. For autosomal dominant in males, females, whoever is affected must be regarded as heterozygous, polydactyly, for example. For autosomal recessive traits, who is normal should be regarded as homozygous normal. For X-linked dominant females who are affected should be regarded heterozygous. Unless you have the information as to that in this woman, both mom and dad are affected, then there are chances for this woman to be homozygous dominant for an X-linked dominant trait. So those are some exceptions. Usually these are rare conditions, so you have to consider these individuals who are affected with a dominant trait heterozygous. Yes?
Can you just repeat what you just said, when to assume if it's homo or heterosexual with the X-linked and all that, like when you're in which scenario?
In terms of X-linked dominant or
just like all of them?
All of them.
Just so we can...
What we got, these individuals? Sure.
Yeah.
X-linked dominant for females, you have to consider whoever, a female, affected with a trait to be heterozygous, just one abnormal allele on one of the X chromosomes. Unless you know that both of the parents are themselves affected or could be affected because of incomplete penetrance. So there are chances that this woman has gotten two abnormal alleles from both of the parents. I think you are confusing yourself in these cases. Just think about the rarity, unless the prevalence or frequency of the abnormal allele is given to you, or consanguinity is involved. Other than that, always consider a person who is affected with an autosomal dominant trait heterozygous, who is normal for autosomal recessive to be homozygous normal, not a carrier, and for X-linked dominant for females heterozygous again. Got it? And for X-linked recessive for females also, you have to consider them as to be homozygous recessive, exactly like autosomal recessive. So, in this case, these are the genotypes of these two guys. So, in order to calculate the chance of the child, two traits are regarded. So, it doesn't say that these genes are linked or so. So, basically, this is what I said. If otherwise unless otherwise stipulated, when two genes are involved, consider them not to be linked. So these are supposed are non-linked genes or unlinked genes. So you can do Punnett square for them separately. For gene T, this is the guy and this is the woman. So autosomal. What is the chance that the child would be affected? This is autosomal recessive. The chance to be affected
50.
50%. So for G